Good afternoon, dear students. Today we have practical lesson number 2. The theme of this lesson will be general and medical protozoology, representatives of the class Sarcodina and type Ciliates. Here on this slide you can see key questions of this lesson. First of all, we need to talk about general characteristics of the class Sarcodina. The main feature of the class Sarcodina is their ability to form pseudopodia or pseudopods, which are outgrowths of the cytoplasm, which serve to move and food capture. Movement organelles determine the division into types. Over 80% of all modern Sarcodina are sea creatures. Some species live in fresh water, few were adapted to life in soil. There are also parasitic species. Sarcodina, which means plasmatic organisms, represent the largest class of protozoa, including more than 11,000 species with a wide variety of forms. Sarcodina have inherent ability to change the shape of their body all the time, which is associated with the formation of pseudopods. Previously, their inconsistency of body shape was explained by the absence of a cell membrane, which couldn't be practically distinguished under an optical microscope. However, using an electron microscope, we can detect the thinnest membrane or plasmalemma, which has a fibrillar structure. The cytoplasm of protozoa forms two layers, the outer, denser ectoplasm and the inner, more liquid endoplasm. In their course, these layers easily pass into each other during their life cycle. Plasmalemma is also capable to be disrupted and to restore. Apparently, this impermanence of layers of the cytoplasm and the absence of a dense, stable membrane determine the uncertainty of the body shape, which is characteristic of Sarcodina. Many Sarcodina form an internal skeleton or an external skeleton in the form of the shells. The organelles of movement are pseudopodia or pseudopods, which are temporary plasma outgrowths of various shapes. It's not only organelles of movement, but also organelles of food capture. Many swimming forms of pseudopodia can only be formations for hunting. They feed on bacteria, algae, and different protozoa. Digestion of captured food occurs in special organelles, digestive vacuoles, which are formed in the cytoplasm. These are bubbles filled with liquid as well as digestive enzymes secreted by the cytoplasm around food particles. Freshwater sarcodines usually also have excretory organelles, contractile vacuoles. Amoebas are mainly inhabitants of freshwater ponds. They are also found in the seas in microreservoirs of riparian sand and soil. In soil, amoebas along with other protozoa are found in an active state. They feed on soil bacteria. Several different species of amoeba can be found in fresh water, on plants, on rotten leaves, and various underwater objects, including the particular large amoeba proteus. Here on this slide you can see this organism, amoeba proteus. This microorganism lives in fresh water, small ponds, and sealed ditches. Moreover, microorganisms multiply easily in laboratory conditions. This is the largest representative among the existing protozoa. During the process of moving, amoeba can grow up to half millimeter and can be seen even without microscope. If the researcher will oversee the amoeba through a microscope, he will be able to see several long pseudopods reminding blades. Outwardly, the amoeba proteus reminds a small gelatinous nubbin. An independent unicellular organism contains a cytoplasm covered with a cell membrane. The outer layer of the cytoplasm is transparent and more dense. Its inner layer is grainy and more fluid. In the cytoplasm there are nucleus and vacuoles, digestive and contractile. By moving, the amoeba slowly flows along the bottom. First of all, it forms a protrusion in some place of the body. It calls a pseudopod. It is fixed at the bottom, and then the cytoplasm slowly moves into it. By releasing pseudopods in a certain direction, the amoeba grows with a speed of up to 2 mm per minute. The body shape of Sarcodina is constantly changing. Part of the pseudopody is getting involved, and some on the contrary, are getting lengthened. The pseudopods are fixed in the soil at several points. It helps the amoeba to move. If amoeba will face with food organics, then it will enfold the entire particle and put it inside the plasmalemma along with liquid. Thus, food bubbles are formed in the body of amoeba, which are called digestive vacuoles, and in these vacuoles takes place the process of organics digestion. The amoeba feeds on bacteria, unicellular animals, and algae, and small organic particles, the remains of dead animals and plants. Around the prey, a digestive vacuole is formed, in which the food will digest and from which it will absorb into the cytoplasm. After this happens, the digestive vacuole moves to the surface of any part of the body of amoeba, and the undigested content of the vacuole will be thrown out. It takes from 12 hours to 5 days for the amoeba to digest food with a single vacuole. In addition, periodically a transparent bubble appears in the cytoplasm of amoeba. This is a contractile vacuole that performs important vital tasks for the microorganism. It stores water from the cytoplasm by increasing in size, and then shrinks it and throws it out through the hole. This whole process takes place during from 5 to 8 minutes. In the body of the amoeba, the amount of organic and inorganic substances is higher than in the surrounding liquid, so water enters into the protoplasm of the microorganism. Due to the contractile vacuole, its excess is thrown out. If this didn't happen, the amoeba would simply dissolve in the water. Together with the liquid, metabolic products are released. That is to say, the contractile vacuole takes part in the process of excretion. With water, oxygen enters into the microorganisms, so organelles help to protozoa organisms to breathe. As in any cell in the body of amoeba, there is a nucleus which is practically invisible. Therefore, experts use dyes to detect it. In the cytoplasm of the amoeba, there is one contractile or pulsating vacuole. It periodically collects soluble harmful substances that are formed in the body of the amoeba in the process of vital activity. Once every few minutes, this vacuole is filled and, having reached its maximum value, approach the surface of the body. The content of the contractile vacuole is pushed out. Except harmful substances, the contractile vacuole removes excess water from the body of the amoeba, which enters from the environment. Since the concentration of salt and organic substances in the body of the amoeba is higher than in the environment, water constantly enters the body, so without its excretion, the amoeba could burst. The amoeba breathes oxygen dissolved in water, which penetrates the cell. Gas exchange occurs through the entire surface of the body. Reproduction of the microorganism occurs in the only way, by segmentation into two cells provided free movement. It occurs through the center of the nucleus, which splits into two halves. The periodicity of this process depends on the temperature of the habitat, nutrition, and occurs once every 2 days. Like all animals, the amoeba has irritability. That is to say, it reacts to signals coming into its body, responds to the impact or irritation of the environment. The amoeba recognizes various microscopic organisms that serve as its food. It creeps away from bright light, mechanical irritation, and increased concentrations of substances dissolved in water, for example, from crystals of salt. What about ciliates? The ciliates are a group of protozoans characterized by the presence of hair-like organelles called cilia, which are identical in structure to eukaryotic flagella, but are in general shorter and present in much larger numbers, with a different undulating pattern than flagella. Cilia occur in all members of the group, although the peculiar sectoria only have them for part of their life cycle. And they are variously used in swimming, crawling, attachment, feeding, and sensation of these organisms. Ciliates are an important group of protists, common almost anywhere there is water, in lakes, ponds, oceans, rivers, and soils even. About 4,500 unique free-living species have been described, and the potential number of extant species is estimated at from 27,000 to 40,000 species. Included in this number are many ectosymbiotic and endosymbiotic species, as well as some obligate and opportunistic parasites. Ciliate species range in size from as little as 10 micromillimeters in some colpodans to as much as 4 mm in length in some gelids, and includes some of the most morphologically complex protozoans. In most nuclei. Unlike most other eukaryotes, ciliates have two different sorts of nuclei: a tiny diploid micronucleus, which is called the generative nucleus, which carries the germline of the cell, and a large polyploid macronucleus, the vegetative nucleus, which takes care of general cell regulation, expressing the phenotype of the organism. The latter is generated from the micronucleus by amplification of the genome and heavy editing. The micronucleus passes its genetic material to offspring but doesn't express its genes. The macronucleus provides the small nuclear RNA for vegetative growth. Division of the macronucleus occurs by amitosis, and the segregation of the chromosomes occurs by a process whose mechanism is unknown. This process is not perfect, and after about 200 generations, the cell shows signs of aging. Periodically, the macronuclei must be regenerated from the micronuclei. In most, this occurs during conjugation. Here, two cells line up, the micronuclei undergo meiosis, some of the haploid daughters are exchanged, and then fuse to form new micronuclei and macronuclei. Cytoplasm. Food vacuoles are formed through phagocytosis and typically follow a particular path through their cell as their contents are digested and broken down by lysosomes, so the substances the vacuole contains are then small enough to diffuse through the membrane of the food vacuole into the cell. Anything left in the food vacuole by the time it reaches the cytoproct anal pore is discharged by exocytosis. Most ciliates also have one or more prominent contractile vacuoles, which collect water and expel it from the cell to maintain osmotic pressure, or in some function to maintain ionic balance. In some genera, such as Paramecium, these have a distinctive star shape, with each point being a collecting tube. Specialized structures in ciliates. Cilia are arranged in rows called kinetes. In some forms, there are also body polykinetids. For instance, among the spiratrix, where they generally form bristles called cirri. More often, body cilia are arranged in mono and dikinetids, which respectively include 1 and 2 kinetosomes, basal bodies, each of which may support a cilium. These are arranged into rows called kinetes, which run from the anterior to posterior of the cell. The body and oral kinetids make up the infraciliature, an organization unique to the ciliates and important in their classification, and include various fibrous and microtubules involved in coordinating the cilia. The infraciliature is one of the main components of the cell cortex. Other are the alveoli, small vesicles under the cell membrane that are packed against it to form a pellicle maintaining the cell shape, which varies from flexible and contractile to rigid. Numerous mitochondria and extrasomes are also generally present. The presence of alveoli, the structure of the cilia, the form of mitosis, and various other details indicate a close relationship between the ciliates, apicomplexa, and dinoflagellates. These superficially dissimilar groups make up the alveolates. Most ciliates are heterotrophs, feeding on smaller organisms such as bacteria and algae, and detritus swept into the oral groove, which is mouth, by modified oral cilia. This usually includes a series of membranes to the left of the mouth and a peroral membrane to its right, both of which arise from polykinetids, groups of many cilia together with associated structures. The food is moved by the cilia through the mouth pore into the gullet, which forms food vacuoles. Feeding techniques vary considerably, however. Some ciliates are mouthless and feed by absorption or osmotrophy, while others are predatory and feed on other protozoa and in particular on other ciliates. Some ciliates parasitize animals, although only one species, Balantidium coli, is known to cause disease in humans. Reproduction. Ciliates reproduce asexually by various kinds of fission. During fission, the micronucleus undergoes mitosis, and the macronucleus elongates and undergoes amitosis, except among the karyorelictan ciliates, whose macronuclei do not divide. The cell then divides in two, and each new cell obtains a copy of the micronucleus and the macronucleus. Typically, the cell is divided transversely, with the anterior half of the ciliate, the proter, forming one new organism, and the posterior half, the opisthe, forming another. However, other types of fission occur in some ciliate groups. These include budding, the emergence of small ciliated offspring or swarmers from the body of a mature parent, strobilation, multiple divisions along the cell body producing a chain of new organisms, and palintomy, multiple fissions usually within a cyst. Fission may occur spontaneously as part of the vegetative cell cycle. Alternatively, it may proceed as a result of self-fertilization, autogamy, or it may follow conjugation, a sexual phenomenon in which ciliates of compatible mating types exchange genetic material. While conjugation is sometimes described as a form of reproduction, it is not directly connected with the reproductive process and doesn't directly result in an increase in the number of individual ciliates or their progeny. Conjugation. Ciliate conjugation is a sexual phenomenon that results in genetic recombination and nuclear reorganization within the cell. During conjugation, two ciliates of a compatible mating type form a bridge between their cytoplasms. The micronuclei undergo meiosis, the macronuclei disappear, and haploid micronuclei are exchanged over the bridge. In some ciliates, which are, for example, peritrix, conatrix, and some suctorians, conjugating cells become permanently fused, and one conjugant is absorbed by the other. In most ciliate groups, however, the cells separate after conjugation and both form new macronuclei from their micronuclei. Conjugation and autogamy are always followed by fission. In many ciliates, such as Paramecium, conjugating partners, which calls gamonts, are similar or indistinguishable in size and shape. This is referred to as isogamontic conjugation. In some groups, partners are different in size and shape. This is referred to as anisogamontic conjugation. In Sesoperitrix, for instance, one sexual partner, the microconjugant, is small and mobile, while the other macroconjugant is large and sessile. In Paramecium caudatum, the stages of conjugation are as follows. Compatible mating strains meet and partly fuse. Then the micronuclei undergo meiosis, producing 4 haploid micronuclei per cell. 3 of these micronuclei disintegrate. The 4th undergoes mitosis. The 2 cells exchange a micronucleus. Then the cells separate. The micronuclei in each cell fuse, forming a diploid micronucleus. Mitosis occurs 3 times, giving rise to 8 micronuclei. 4 of the new micronuclei transform into macronuclei, and the old macronucleus disintegrates. And then, at final stage, binary fission occurs twice, yielding 4 identical daughter cells. What about DNA rearrangements or gene scrambling? Ciliates contain two types of nucleus, somatic macronucleus and the germline micronucleus. Only the DNA in the micronucleus is passed on during sexual reproduction, conjugation. On the other hand, only the DNA in the macronucleus is actively expressed and results in the phenotype of the organism. Macronuclear DNA is derived from micronuclear DNA by amazingly extensive DNA rearrangement and amplification. Development of the oxytricha macronuclear genome from micronuclear genome. What is it about? The macronucleus begins as a copy of the micronucleus. The micronuclear chromosomes are fragmented into many smaller pieces and amplified to give many copies. The resulting macronuclear chromosomes often contain only a single gene. In Tetrahymena, the micronucleus has 10 chromosomes, 5 per haploid genome, while the macronucleus has over 20,000 chromosomes. In addition, the micronuclear genes are interrupted by numerous internal eliminated sequences. During development of the macronucleus, internal eliminated sequences are deleted and the remaining gene segments, macronuclear distinct sequences, are spliced together to give the operational gene. Tetrahymena has about 6,000 internal eliminated sequences, and about 15% of micronuclear DNA is eliminated during this process. The process is guided by small RNAs and epigenetic chromatin marks. In spirotric ciliates, such as oxytricha, the process is even more complex due to gene scrambling. The macronuclear distinct sequences in the micronucleus are often in different order and orientation from that in the macronuclear genes, and so in addition to deletion, DNA inversion and translocation are required for unscrambling. This process is guided by long RNAs derived from the parental macronucleus. More than 95% of micronuclear DNA is eliminated during spirotric macronuclear development. These organisms are the most complex protozoa. Ciliates can be segregated and colonial by type of food, saprophags, predators, and parasites. The movement is carried out with the help of cilia. Usually they have several nuclei which differ in structure and function. The cell is covered with the pellicle, so the body shape is constant. The organelles of attack and defense are trichocysts that cover the entire surface of the body of the ciliates. You can see these organelles here on this slide. Ciliates perceive various external irritants and react accordingly to them. As a rule, the response to irritation is their spatial movement. The irritation may cause a purposeful movement to or from the source of the irritation. When the ciliate moves towards the irritant, they talk about a positive taxis, and if it avoids it, about a negative one. Paramecium caudatum lives in stagnant water ponds. It has two nuclei. The large polyploid nucleus, macronucleus, is responsible for vegetative functions, nutrition, respiration, and excretion. The small nucleus, which calls micronucleus, is involved in the sexual process. The movement is carried out with the help of numerous, more than 10,000 cilia located in regular longitudinal rows. They make coordinated undulatory movements. The power supply is as follows. On one side of the body there is an infundibular deepening leading to the cellular mouth and the tubular pharynx. With the help of cilia, paving the funnel, food particles, bacteria, single-celled algae, detritus are driven into the mouth and then into the throat. From the pharynx, food by process of phagocytosis enters into the cytoplasm. The resulting digestive vacuole is picked up the circular current of the cytoplasm. During from 1 to 1.5 hours, the food is digested, absorbed into the cytoplasm, and the undigested remains are removed through the hole in the pellicle, the cytoproct. On the images of ciliates in the front and back part, you can see a paired internal star-shaped organ, a contractile vacuole with ampoules located around it. The concentration of dissolved substances in the body of ciliates is higher than in the environment, and due to the difference in osmotic pressure, water rushes inside. The liquid first fills the ampoules, which at some point are emptied into a contractile vacuole that inflates into a bubble. Then, through a special channel, the water is thrown out. Ciliates have two contractile vacuoles, they pulsate and alternately. Asexual reproduction. First, both nuclei are divided by mitosis, and then the body of the ciliate is divided on halves in the transverse direction. The missing organelles develop anew in both daughter individuals. Sexual process. Conjugation. In this case, the two ciliates are applied to each other by the sites where the mouth is located. The pellicle at the point of contact of individuals dissolves, and a cytoplasmic breach is formed between them. Then small nuclei divide meiotically. The large nucleus is destroyed and doesn't take part in the sexual process. Of the 4 haploid nuclei which are formed in each ciliate, 3 disintegrate. Then the remaining 4th nucleus divides mitotically one more time. One of the 2 formed nuclei, stationary, remains in the same ciliate, and the other, migrating, passes into the cell on the conjugation partner. After the fusion of the stationary and migrating nuclei, a diploid nucleus with recombined genetic material is formed. In each of the ciliates, the diploid nucleus divides several times, and after a series of transformations, a small and renewed large nuclei are formed. After a while, the paramecium caudatum begin active asexual reproduction by division. Paramecium caudatum uses 2 methods of reproduction, asexual and sexual. So this organism is a component of the biotic community in the food chain, and also ciliates and other protozoa organisms are living in the soil help to increase the fertility of irrigated lands in the southern regions. And now about parasitic or pathogenic representatives of these classes. Some protozoa species can lead a parasitic lifestyle, settling in the intestines of vertebrates and invertebrates. In humans, 5 types of microorganisms live in the intestinal cavity. 4 of them are considered harmless, but the 5th, which is called dysentery amoeba, can cause a serious disease amoebiasis. This type of protozoa is a very small microorganism that live in the large intestine. Their main characteristic is considered to be high mobility. Amoeba exist in almost all parts of the world, but the disease amoebiasis is quite rare, only in the southern and warm regions. This is due to the fact that presence of a parasitic microorganism is not always accompanied by a disease. In this case, the dysentery amoeba performs the function of a carrier, that is, it moves in the intestinal environment, feeds on bacteria, but doesn't cause pathological changes. But sometimes the microorganism actively begins to penetrate the intestinal wall. This action causes a person to have a severe form of bloody diarrhea. During this period, the amoeba changes its food object, starting to eat red blood cells instead of bacteria. If you do not undergo a course of treatment, then amoebiasis will turn into a chronic form, causing severe depletion in a person. Dysentery microorganisms can only live in the human body, and when they get into the soil or water, they immediately die. Infection is carried out by non-permanent forms of existence, cysts. When protozoa from the large intestine enter the rectum, they change their shape. They draw in the pseudopods throughout food particles and become round, and the ectoplasm forms a thin but durable coating. This process is called encystin. A feature of this moment is the division of the nucleus successively twice, forming characteristic quad core cysts. In this form, they are excreted with feces, where they continue to exist for from 2 to 3 months. If the parasitic protozoa organism enters the human body, it starts its life cycle in new host. Sectoria. They are characterized by the absence of flagellums, mouth, and pharynx in the adult state. Feeding occurs using thin tubular fillers. Parasitic Infusorium Ballantidum coli causes diseases of animals and humans, colitis of the intestine. Fish are often affected by the Infusorium ichthyopteros. It penetrates the skin, fins, and gills of fishes, leading to their death. Thank you for your attention. Have a good day.